So now to the business at hand. So two excellent 30-minute talks today. Our first speaker is Dr. Lucia Jilaveanu. Lucia, as many of you know, has been a postdoctoral fellow in the laboratory of Dr. Harriet Kluger. She received her medical degree in Romania, her PhD at Wesleyan University, and her work has really been focused principally on the biology of melanoma, but through her work with Harriet has evolved into the sort of understanding the biology of brain metastasis, clearly an important issue, particularly for melanoma, but for a variety of malignancies. And I think understanding the biology and how to target brain metastasis is clearly an evolving field that we're trying to improve for the benefit of our patients. Beyond her success in publications, Lucia was recently awarded a R01 grant to support her research in her laboratory, which is an enormous accomplishment. And we're pleased to hear from her today. Her talk is entitled, as you see, New Molecular Targets for Melanoma Brain Metastasis. Lucia, thank you.
Thank you so much for the introduction, and thank you for having me here today and giving me the opportunity to introduce some of our work. And I will start with by introducing the clinical relevance of our work. Brain metastases are the most intracranial tumors in adults. They account for more than 50% of brain tumors. In patients with disseminated disease, brain metastases occur in up to 30% of cases. We have seen a rise in the incidence in brain metastases, but this is probably due to having better detection methods and improved survival of metastatic patients. In terms of the primary lesion, lung cancer has the highest incidence with a proportion of 20%. Melanoma has the second highest incidence with 6.9% and is closely followed by renal cell carcinoma with 6.5%. While lung cancer and breast cancer are more prevalent, melanoma has the highest propensity to metastasize to the brain. Almost 40% of stage 4 patients are found to develop brain metastases during their lifetime, and brain metastases are actually found at autopsy in almost 75% of cases. Median survival used to be very low, in the order of a couple of months. New series report a range of 8 to 11 months. But ultimately, brain metastases are responsible for the approximately 50% of all melanoma deaths. Current treatment options include surgery, radiosurgery, whole brain radiation therapy, and in the past couple of years, we have seen a lot of progress in the development of therapies with activity in the brain. But median progression-free survival continues to be in the order of months. More recent trials of approved systemic therapies include combinations of dabrafenib and trametinib with response rates in the brain of ranging from 44 to 59%. Our Yale phase 2 trial of pembrolizumab and combinations of ipilimumab and nivolumab, which recently was reported to show a response rate of 54% in the brain. Experimental therapies include inhibitors of CDK4, combinations of pembrolizumab and bevacizumab, and nivolumab with IDO inhibitors. But despite the progress that has been made, systemic therapy for patients with melanoma brain metastasis continue to be limited. First, because these patients are excluded from most clinical trials if they have active brain disease, particularly in the early phases of the development of the drugs. But secondly, because the development of new drugs has been restricted, and this is due to having limited studies of drug penetration, and also because of our lack of understanding of the biology of melanoma brain metastasis. So there is critical need for research to focus on the pathogenesis of and treatment of melanoma brain metastases, and these are the areas on which my research focuses on. And regarding the pathogenesis of brain metastases, there are many questions to which we continue to have limited answers. And I started off by saying that among cancers who metastasize to the brain, melanoma has the highest propensity. But what drives melanoma propensity to spread to the CNS? And there are more specific questions like, is brain tropism in melanoma preprogrammed? And this idea of cancer cells being hardwired to metastasize to specific organs is more than a century ago old. But whether or not this brain tropism is a characteristic of select cells at the primary tumor level, or is something that's fully acquired much later on after local or distant metastases are formed, is still unknown. What are the molecular determinants of melanoma brain metastasis? What are the underlying mechanisms? Are these unique? And there have been observations in other diseases showing that melanoma brain metastases do harbor unique molecular features, but is this the case for melanoma? Can we find commonalities with other diseases? And last, does the host play a role? What is the role of the host? Does it suppress or does it facilitate brain metastases? And regarding our first question that relates to this pre-existent tropism, I would like to mention here a couple of very interesting observations we made toward the more early steps in our studies. And the first observation comes from our sequencing studies that showed that genes more frequently mutated in early brain metastasis converge to genes that are highly expressed in the normal brain. And this is what you can see here, an analysis in the upper panel in red, various locations throughout the CNS. The analysis was done with frequently mutated genes in early brain metastasis and then a random selection. Our second observation comes from our transcript profiling studies of extracerebral metastasis, again showing that in early brain metastasis there is upregulation of genes that have a known role in neural cell development or associated with normal neural function. So these two very interesting observations helped us frame our hypothesis around this idea that brain tropism might be pre-programmed in melanoma. Relating to the question about the host, whether or not it suppresses or facilitates brain metastasis, to date we don't have enough studies here. We, for example, show that patients with highly inflamed tumors are less likely to have tumor dissemination to the brain. And this is what I'm showing in this Kaplan-Meier curves. Significant association between high density of CD3 and CD8 TILs in extracerebral metastasis with increased time to developing brain metastases. But there are other studies that focus more directly looking at the tumor microenvironment in the brain, showing that the host-tumor interactions can actually favor brain metastatic progression, and the most compelling example being the supportive role of the astrocytes in melanoma growth. So brain metastasis might be mediated in part by tumor microenvironment related factors. Mechanisms are, however, unknown and probably complicated. And this takes me to our overall objectives. So our goals were are to identify molecular networks involved in melanoma brain homing, to uncover potential drivers of melanoma brain metastases, and among those to possibly identify novel molecular targets that could be harnessed for drug discovery and then used for treatments of patients with brain metastases in melanoma. And last but not least, to better understand the pathogenic mechanisms of in our disease. So this is just a short overview of the type of resources we used in the studies, and we combined both computational approaches and experimental approaches. And this type of integrative approach has been used extensively in research, particularly in the area of molecular target discovery. But our studies were initially focused on molecular profiling a large cohort of metastatic melanoma tumors and patient-derived cell lines, and we used here genomic and transcript profiling, and also combined data from protein profiling and tissue microarrays to eventually integrate studies of cell line models and mouse models. And I will take a minute and discuss these. So preclinical models of brain metastases in our disease have been limited, and to facilitate the studies, we set out to develop new models. In our models, brain metastases are induced by injection of cells in the left ventricle of the heart. And this method is a very well-established method that was shown to allow selection of clones and subclones with different organotropism. And so the cells are injected in immunodeficient mice, and since they're dispersed systemically, they have equal opportunities to reach and grow in different distant organs and form metastases or remain dormant. Eventually, we recover these cells and culture them, and then re-inject in several cycles until we obtain a derivative that has increased predilection for the brain, meaning it induces brain metastases most frequently and with high incidence. And then this cell line can be then compared to the parental cell line for hints on mechanism and also in functional studies. And we have developed several models using genetically diverse melanomas. Most of our models produce parenchymal lesions. So going back to our study, we took a funnel approach, starting with discovery studies where we transcript profiled over 100 patient specimens, tumors from patients with brain metastases in brain lesions, and analyzed this data against data that we obtained from our cell line models of brain metastases to be able to select genes that specifically associate with brain metastasis. And since RNA and protein correlate only about half of the times, in the next step in validation studies, we're using tissue microarrays and quantitative immunofluorescence, looking at the association between protein level and brain propensity. And some of these molecules are also studied in vitro to get hints on their function, and we're using different cell-based assays here. Eventually, the most interesting molecules we're analyzing in vivo using different mouse models. And so this approach allowed us to identify several very interesting molecules that my lab is now focusing on characterizing more in detail. But I would like to focus the rest part of my talk on one molecule in particular that we identified via this pipeline called PLEKHA5. So this molecule is a member of the plextin homology domain containing family A. It was also called PEP2. There are two well-distinguished splice variants. Both of them have been involved in different stages of the brain development. The protein is expressed at the plasma membrane, but also associated with microtubules. At the time we identified this molecule, ligands and signal pathways were unknown. So just to summarize our initial findings, PLEKHA5 seemed to be a potential mediator of melanoma brain metastases since we found that it was the most significantly differentially expressed gene in our patient cohort and a cell line model of brain metastases. Using tissue microarrays and quantitative immunofluorescence, we then found that high tumor PLEKHA5 associated with early development of brain metastases, and this is what you can see in this Kaplan-Meier curves, association between high and low expressors with brain metastasis-free survival, which we define as the time from developing the first distant metastasis to brain metastasis by MRI. And so now I will talk a little bit about our in vitro data loss of function studies revealed a role of our protein in proliferation, and this is what you can see in the upper graphs, comparison of proliferation rates between cells that inherently overexpress our protein and cells in which we knock down our protein. So knockdown inhibits proliferation. But then cell cycle analysis showed that this is due to an inhibition of cell cycle transition from G1 to S phase, and this is what you can see in all these graphs in the bottom, where we plotted the number of cells or percent cells in each phase of the cell cycle for comparison. We can see that knockdown reduces the number of cells in the S phase or the DNA synthesis phase in the 3 cell lines. In our control cell line that expresses negligible levels of this molecule, knockdown produces no effects, while this time overexpressing our molecule increases the number of cells in the S phase. And this translated in vivo in differences in tumor growth and tumor formation. So subcutaneous implantation of cells in which we knock down the molecule induced a significant inhibition of tumorogenicity. This is what I'm showing in this upper left panel. Approximately half of our mice, of these mice, are tumor-free in the knockdown variant, and this was plotted here in the graph. But knockdown also induced a delayed tumor growth, and you can see the corresponding graphs from the 3 models we used, differences in growth proliferation curves between wild type or cells that overexpress endogenously our molecule and cells in which we knock down our molecule. And then our control cell line, conversely, we see that overexpressing the protein produces tumors that grow at higher growth rates when compared to controls or knockdowns. But because our studies are focused on brain disease, we wanted to look at this in a more relevant context and found that knockdown of this molecule also significantly inhibited intracranial tumor growth. This is what you can see in these bar graphs in 2 of our models. In these graphs, we plotted average tumor size to compare the difference between the cohorts. In the bottom, third cohort or a third model, differences at microscopic levels. And now turning again to some more in vitro data, which we think it's very exciting. We initially used the RPPA or reverse phase protein arrays, which is a quantitative proteomic approach to look at downstream signaling effects associated with PLEKHA5 modulation across many cell lines, and found a set of molecular changes that consistently converge to PI3 kinase pathway or molecules that interact with PI3 kinase. Eventually, our studies led us to identify one molecule called PDCD4. PDCD4 stands for programmed cell death 4. This protein is a relatively new tumor suppressor that has been scarcely studied in melanoma. We find loss of PDCD4 in more than half of our cell lines and metastatic melanoma tumors. But here I'm showing a Western blot that summarizes results from or signaling changes associated with PLEKHA5 knockdown. Our studies demonstrated that PLEKHA5 is an important suppressor of PDCD4 in melanoma. So here, for example, in one of our cell lines, knockdown of PLEKHA5 induces upregulation of PDCD4, and then when we overexpress PLEKHA5, again, we see down modulation of PDCD4. But programmed cell death 4 is known through its effects on cell cycle as it upregulates P21 and P27, and this has been shown in a couple of studies, causing arrest at G1, which corroborates with our previous findings. So you can see modulation of this important cell cycle factors here. In this case, P27 going up in the knockdown and then going down again in the overexpressing cell line. And of course, the downstream effector, a very important negative regulator of the cell cycle, retinoblastoma protein, whose phosphorylation inhibition associates with the progression of the cell cycle. So you can see differences from the wild type, knockdown, and then overexpression. So then you can see all these modulations very nicely in other cell lines following knockdown of PLEKHA5. But how is PDCD4 regulated? In other diseases, the main mechanism has been shown to be related to microRNA suppression or promoter hypermethylation. Our studies show that in melanoma, the main mechanism is targeted proteasomal degradation. And this seems to be facilitated by PLEKHA5. And I put this diagram together to show you some of the pieces of the puzzle that we recently were able to demonstrate strongly with our studies. And also some of the things that we're currently still investigating. For example, we know for sure that PDCD4 requires to be phosphorylated in order to be targeted to degradation. We know that AKT and mTOR are important for this phosphorylation. We also know that PLEKHA5 acts downstream of this event, but we don't know exactly how it facilitates final degradation of the protein. And we're also investigating other interesting things like feedback loops back to the PI3 kinase pathway. As we're trying to better understand this novel regulatory mechanism in our disease. But how does our work fit with the in the current field? Our work is in line with studies showing that PI3 kinase pathway might be more important, or signaling might be more important in cerebral metastasis than extracerebral metastasis. Higher degree of pathway activation was found in brain metastasis when compared to extracranial lesions from the same patients, but mechanisms of pathway hyperactivation have not been studied sufficiently. PLEKHA5 might be a link between PI3 kinase signaling and brain metastasis, so PI3 kinase pathway inhibition via PLEKHA5 might be an effective strategy in our disease. But what is our clinical experience with drugs that inhibit this pathway? Monotherapy showed limited success in metastatic melanoma due to acquired resistance and insufficient blockade. But factors involved in this resistance have not been studied. Strategies to overcome feedback loops and pathway crosstalks are needed, and so identifying molecules like this molecule that interact with PI3 kinase is important. And finally, some evidence, but this is preliminary data, that supports the notion that suppression of PLEKHA5 might increase sensitivity to PI3 kinase inhibitors. So we find that when we inhibit the upstream activator of AKT, PI3 kinase with RLY or BKM, or its downstream target mTOR with rapamycin using suboptimal concentrations of drugs, results in a more efficient inhibition of pathway signaling in cells in which we suppress the PLEKHA5. And we're measuring as a surrogate the levels of phosphorylated S6 protein, which is the most downstream or more downstream effector in the pathway. So this can be seen, for example, in the cell line, the levels of inhibition of S6 protein in cells by LY and rapamycin, cells that endogenously express our protein, compared to cells in which we knock down our protein. So partial inhibition here compared to no treatment, and very effective inhibition here compared to no treatment. And this was also validated in another cell line. So PLEKHA5 targeting might provide an effective and vertical pathway inhibition. And drugs that target this pathway are currently in clinical development. Here are the 6 ongoing trials that include inhibitors of PI3 kinase AKT signaling. None of these ongoing trials allowed enrollment of patients with melanoma and brain metastasis. And this takes me to the conclusions of our study so far. So we conclude that brain tropic melanomas are biologically distinct. Our studies also support the idea that molecular aberrations associated with brain tropism can be seen at extracranial locations. As an example, as it relates to today's talk, we found a molecule that is aberrantly expressed in melanoma brain metastasis, but we have other molecules that we're currently investigating. There are multiple pathways and pathway crosstalks that are probably involved. This is what our data shows, and positive and negative regulators should be looked at since they might play a role. For example, in this case, PLEKHA5 seemed to be a regulator of melanoma growth via PDCD4 in brain metastasis. Ultimately, the overall conclusion is that uncovering specific pathways in brain metastasis will lend support for the development of drugs that target this specific pathway. So PI3 kinase, for example, targeting drugs in conjunction with PLEKHA5 inhibitors should be explored. And of course, there are many people to acknowledge here, starting with members of my lab, Hongyi Zhang and Hui Fang, who has been recently started to work on this project, and others who had a more supportive role, but more importantly, my mentor, or as important, my mentor, Harriet. I worked with Harriet for many years, first during my training and now in collaboration. Harriet has been a tremendous help and excellent mentor. And of course, the Sport Tissue Specimen Core, who has been supportive of this work, and many collaborators that participated and helped me throughout the studies, the funding sources, and thank you everybody for listening.
Thank you. So questions for Lucia? Well, I'll start. So, one thing that you obviously have to sort out is there are features about some of these molecules that will promote proliferation and maybe other aggressive biology that might be associated with brain tropism, but not specific because they just preserve a much more aggressive biology. How do you separate that?
With mouse models. So that's why we had developed different mouse models of diverse genetic melanomas to be able to sort through those more specific molecules that associate with brain metastasis.
Right.
Yeah, two quick questions. Do you think that PLEKHA5 could be used predictively, so you could, when you see a patient, use that, perhaps say who's at more risk for brain metastases and alter the treatment somehow?
Yes, I think so. Whether or not we can look at the primary tumor level, we don't know. We haven't looked there, since in melanoma there is a big gap between primary disease and metastasis in terms of timing. But for metastasis, yes, I think it could be a good marker to look at.
My second part, is this melanoma specific? Have you looked at this in any other tumor types?
We have not looked in other tumor types, no, but that would be interesting.
Any other questions? Well, again, thank you.
Thanks.
So our second speaker for the hour is Sangini Sheth, and she is an assistant professor of obstetrics, gynecology, and reproductive sciences at Yale. Sangini was a graduate of Yale College and received her MD and MPH at Johns Hopkins before returning to the faculty here at Yale. And beyond being an expert across multiple domains in gynecology, she obviously has been leading an effort to improve the compliance and effectiveness of HPV vaccination and prevention of cervical cancer. So, Sangini, thank you. I'm just going to try to pull your slides up here. You may be better at this than I am.
Oh, yeah. Let's see.
I guess it's not just me. Okay.
Good afternoon, everyone. Thanks for the introduction. So this is a little bit of a change of pace on the other spectrum of where research can lie. I have one disclosure. I've received drug-only HPV vaccine from Merck for this research. Just as an outline of what I'm going to cover today, I'm going to go over a background on HPV-related diseases and the HPV vaccine, including current immunization rates and barriers to vaccination. I'm going to do a brief introduction about the role of implementation science and kind of key characteristics of this area of research, and then jump into our particular strategy that we've been investigating over the last year and a half, trying to vaccinate a specific group of women, and then and how we've applied implementation science methodology to that. My goals for all of you as you walk out of here this afternoon are to be aware of the barriers to HPV immunization, to understand the role and relevance of implementation science, and to be able to discuss the strengths and weaknesses of a particular strategy of inpatient postpartum immunization. So as an overview of HPV disease burden in women, and this is really focusing primarily on cervical burden. Of course, HPV-associated diseases can affect and is associated with vaginal and vulvar cancer, with anal cancer, with oropharyngeal cancer, and in men with penile cancer. But kind of digging a little bit deeper into disease of the cervix, the tip of the iceberg is cervical cancer, with about 4,000 cervical cancer deaths in the U.S. annually. But then there's a whole range of other precancerous lesions that we should be focused on trying to reduce and eliminate as well, which an HPV vaccine can help us do. So there's 330,000 new cases of high-grade precancerous dysplasia every year, 1 million new cases of genital warts, and 1.4 million new cases of low-grade cervical dysplasia, which also, has a significant financial burden on our healthcare system. So moving over to the vaccine itself, so there has been an HPV vaccine available in the U.S. For over a decade now, since 2006. The current recommendations are for routine vaccination to all 11 to 12-year-olds, and then looking specifically at the recommendations for females to continue to vaccinate through age 26 for those who have not already completed the series. And more recently, the vaccine schedule was modified so that for adolescents under 15, there's a 2-dose series, and then for 15 and older, we remain at a 3-dose series. There is currently, since 2014, we have access to a 9-valent HPV vaccine, so that means it's protecting against the 2 HPV strains that are known to cause 90% of genital warts, but also 7 high-risk or oncogenic HPV strains that are associated with 90% of all HPV-associated cancers and specifically 80% of cervical cancers. And these are vaccines that have been shown to be highly efficacious in preventing this whole spectrum of disease. So, sounds great, right? We have a clinical problem, we have a preventative vaccine. Unfortunately, we are not doing very well in terms of uptake of the vaccine. So what this graph shows is in dark, this is data for 13 to 17-year-old girls in the United States. So this top line is the rate of Tdap uptake. So that's another vaccine that's recommended for 11 to 12-year-olds. And we see that it's very high. And then right below it is uptake of meningococcal vaccine, another vaccine recommended for 11 to 12-year-olds, also very high. And then lagging far below that is uptake of the HPV vaccine. So this line right here is uptake in girls, and that's getting 1 dose on board. We're currently at about 60%. But up until recently, it was a 3-dose series for everyone. And so when you look at rates of getting all 3 doses on, the numbers drop off significantly. So we're at less than 50% when you start to look at completion of the series. This is national data. If you look at Connecticut data, it's very similar. We're in the same situation. So what are the barriers? Why is this not happening? There have been lots of studies looking at this. Probably one of the most important issues here is lack of consistent and timely recommendations from clinicians. Either the recommendation for the HPV vaccine is a little bit softer than recommendations they may give for other vaccines, or it's the recommendation is, you should get it at some point, not you're here today, you're due for it, you should get it today. Or there's some trying to assess a given patient's risk of whether that is a risk of getting cervical cancer down the road, or risk of sexual activity, or risk of sexual transmitted infections, whatever it might be. But we know that we can't look at somebody and assess those kinds of risk factors. Then there's also this concept of anticipatory caution. So being worried at how a patient or a parent might react to discussion of the HPV vaccine, whether that's because you may get into the territory of having to talk about sexual activity, or get into the territory of talking about sexually transmitted infections, or just vaccines in general. And then parents may have some of their own ambivalence. Well, I've heard XYZ about this vaccine, or, well, I'm not sure that my child needs it, or I'm not sure that my child needs it right now. And then, the ever-present concern of side effects related to vaccines. In particular, the HPV vaccine has its own unique set of unfounded side effects that it's been associated with. And then the dosing schedule, the fact that it's a multi-dose regimen, and that especially tied to this issue of completing the series, less so with starting the series at all. And so this is where now implementation science comes in. We have this excellent, highly efficacious, evidence-based recommendation, and it's not being used. So that's the role of implementation science, which is basically a study of the methods to promote the integration of research findings and evidence into healthcare practice and policy by understanding behaviors of healthcare providers, other stakeholders, in a very interdisciplinary sort of way. You're all familiar with the spectrum of research, and so we're really kind of talking about that last piece of the puzzle. So this kind of research is conducted in real-life settings and established health systems. It investigates the bottlenecks that might be in place. It tests new approaches, new strategies to delivering that evidence-based intervention. And it has some interesting features. So it's systematic. So it still has a lot of rigorous methodology associated with it. It's very much multidisciplinary from a whole host of factors, from the science to social issues, economic, political, systems-based issues, and bringing all those people together to understand the process and develop unique strategies. Is very contextual, so understanding and developing strategies within the local context, but still developing generalizable knowledge. And it's also trying to look at the issue, what the barriers are at multiple levels. One thing that people who have heard a little bit about implementation science probably hear about very quickly is this idea of frameworks. So there's lots of frameworks within this field that all have their own special acronyms. Like any conceptual design, the idea of a framework, even in implementation science, is it can help diagnose what the challenges might be. It can help with evaluating an implementation program, and then it can help explain findings or using it as an analytic tool. A more common framework, and one that I like to use, is the Consolidated Framework for Implementation Science. As the name implies, it's actually a consolidation of numerous other frameworks, and basically it is trying to look at any given problem, any given implementation process from the lens of the outer setting, so larger economic, political viewpoint, from the inner setting, so from the local setting, whether that's a particular hospital or a particular community, and looking at the individuals involved, all of these key stakeholders, and the process of implementation, and then how all of these things affect the evidence-based recommendation. So now that I've given a little bit of a brief introduction to implementation science, I'm going to talk about one immunization strategy that we have been looking at, where we're trying to vaccinate inpatient postpartum women. So the idea is basically that a woman delivers, and if she's under the age of 27, which is the group in which the HPV vaccine is recommended, while she's still here in the hospital, we can get a vaccine on board. Why might this work? So the vaccine cannot be given to pregnant women, and so many of these younger women may be engaging for the first time after a few years with the healthcare system in the context of pregnancy, but now there's this 10-month period where they can't get the vaccine. So once they're delivered in the hospital, that's the first opportunity to give it. It is safe to give to breastfeeding women. It will potentially reduce the number of additional visits that they need, so it's a patient-centered approach. Some of these women may have already gotten 2 doses previously, so it's an opportunity to actually complete the series for some of them. Those of them that are starting this series, their second dose is paired with their routine outpatient postpartum visit. We have a lot of experience immunizing women in the postpartum setting with other vaccines. So influenza, Tdap, MMR are all commonly given to women if they're not up to date on them already, shortly after they deliver, before they are discharged to home. And then I think the big question is, well, obviously these women have already been sexually active, so what's the point? And there's actually a huge point, because there's a lot of disease that can still be prevented, even in this age group with that sort of history. We know that we can have up to a 30% reduction in high-grade cervical dysplasia. We can have a significant impact on reducing the risk of genital warts, and we can even reduce the risk of cervical excision procedures. So those are treatments that are necessary in women who have these high-grade precancerous lesions by up to 25%. And there's even data to support that women who previously had high-grade dysplasia who then end up getting vaccinated, there may be an opportunity here for them to prevent future high-grade dysplasia. So there has been some other work looking at this type of a strategy, which is not certainly meant to be a strategy in isolation. It's meant to be, what are all the different ways that we can close up the missed opportunities in delivering the HPV vaccine? This group, Berenson, a few years ago, just did a kind of cross-sectional hypothetical survey among women in a population that had very low vaccination rates, in terms of trying to gauge their willingness to receive the vaccine after delivery in the inpatient setting, and that was, a high degree of willingness, basically. Another group in New York, they looked at specifically women who had never received any doses of HPV vaccine, and looked at uptake of that first dose in the inpatient setting. And that first dose uptake was very high. Their outpatient setting was probably not optimized to be really thinking about the HPV vaccine, and so their completion rate ended up still not being very high. But again, they found that women were very happy with this type of program. They also, there were some other weaknesses to this. We didn't have a comparison group to look at. There wasn't really an explanation of the women who decided not to participate in this, what was going on with them. And then this last group recently published a paper on how effectiveness of this type of a program using patient navigators. So the patient navigators were people who were trained by the study team to do everything from counseling patients to reviewing their immunization records, identifying the ones that were eligible to get the vaccine, to then doing phone calls and text messaging on the outpatient side to really garner a high level of uptake as a research study. And again, they found that 75% of those who needed the vaccine received a postpartum dose, and they found their overall completion of series to increase significantly. So right now we're just conducting pilot studies here, really to establish some baseline information. One is to just determine the feasibility and uptake of this type of a program, and just the implementation of this type of a program here at Yale. And then to, as part of that, evaluate the experiences of both the patients and the providers in adding this sort of a thing to the postpartum setting. So the first piece of this, keeping in mind an implementation science methodology, is engaging stakeholders. And we kind of identified three groups of broad groups of stakeholders. The first was at the hospital level, kind of more broad systems wide, and then second within the hospital, this very specific inpatient postpartum setting, and then the outpatient providers, because patients are kind of moving through all three over the course of the pregnancy. Diving a little bit deeper into the broader hospital level, we had to engage pharmacy. We needed to get the supply of the vaccine, which is available in the outpatient side, but it's not actually on the inpatient formulary. We had to engage Epic in terms of billing our orders and linking to immunization history. And then we had to work with various care providers, specifically at the hospital level, the residents. And then again, same thing, we had to continue to engage pharmacy, we had to develop workflows, we had to orient the postpartum nurses who are not familiar with the HPV vaccine, because it's, again, primarily been an outpatient focused intervention. And then using some tools from the quality improvement world to develop a system that's both working for the providers and for the patients, because we're introducing a new work element for everyone. And on the inpatient side, doing orientations with all the providers so that they can start talking about this type of a program from early on, because the more patients hear about it, if they feel that it's being endorsed by their obstetric providers, the more likely they are to actually utilize the program. Kind of recommending some best practices for our providers, and then providing some patient outreach tools for the patients in English and in Spanish. And then this is just some of the types of data that we were collecting, looking at some feasibility measures, looking at some effectiveness measures, basically to see, what do the numbers look like in terms of women who are eligible, that those that accept, those that successfully actually get the vaccine, and in that process trying to identify barriers. And then we also conducted qualitative studies, again, with the patients and with providers. So we're still in pilot phase. It took a year just to get the program started. And we launched at this campus in April and at the St. Raphael's campus in June. And so to date, we've had 99 women who are under the age of 27. And of those, 57% were eligible to receive the vaccine, meaning the other 43% had completed the series. So among those that were eligible to receive the vaccine, the uptake in that group is 73%. So in this graph, this is the group that has successfully gotten vaccine in the inpatient side, and then this is the group that did not get the vaccine, and the majority of them are women who declined, although there are a few here where the vaccine didn't get ordered for them, or the patient ended up leaving before the dose was actually administered, so more kind of process systems issues there. Things that stand out, there's a significant difference here in terms of language of patients. The vast majority actually were, of those who received the vaccine, had never gotten a dose prior, although we did complete this series for 7% of women because they'd already gotten 2 doses before. In terms of patient interviews and kind of, enriching quotes that we've picked up from talking to patients, the first 2 rows are more about the patients themselves, so knowledge that they may have gained in this process about the vaccine, or their personal views or opinions. So I'll read from the views and opinion row. But I feel like me being here after delivering my children, why not? It's for a good cause, and if it can help, cure things and help avoid risk, then it's good. One other patient speaking to convenience, I chose to do it yesterday. They were there and they had the vaccination right there. And then in terms of the patient-centeredness, this idea of it wasn't too much of a hassle. They explained it to me, but they were still very quick. It's not mandatory. So they felt like they were a decision maker in this process. So we'll get into some of the challenges that we saw along the way. This is actually the challenges that I thought we would encounter, that the clinical need or value of this could get questioned. These are women who have just delivered. They have maybe more pressing things, both clinically and psychosocially, to think about. And so whether that's providers questioning or patients questioning, that it may be too much of an additional work burden on postpartum nursing staff, this idea of being poked and prodded a little bit too much. And actually, none of these played out. The patients have overall been very engaged in this. The nurses and providers have been very engaged in this. They very much see this as advancing women's health, and haven't really pushed back at all, and have even been very patient as we've kind of had to work out kinks along the way, from a practical standpoint. So what we have encountered, and I'm going to go to the barriers column first. As I mentioned, it took us a year to even get this going. There were lots of challenges, and actually continue to be lots of challenges, to getting access to the vaccine on the inpatient side, and that's a really complicated algorithm of Medicaid policy and payers and the cost of the vaccine, and that this is seen as an outpatient intervention. And so right now we're really doing it in a very small pilot fashion for a very small proportion of women. And what I didn't mention when I was showing the data was there's probably another 230 women we could have immunized if we were working on a more expanded level and not just focused on the pilot group of the Yale New Haven Hospital's Women's Center patients. Then, a challenge with vaccines in general is inconsistent immunization records. So if you've been immunized within the health system, then we have those records in EPIC. But if you've been immunized outside of the system, then there's no way to know your immunization status. There's no immunization registry in the state of Connecticut, which some, for older people, which some states have. And so we have to do the best that we can with that. Of course, the cost of the vaccine continues to be a challenge. It's $150 a dose. That's kind of, the market value for that, plus whatever costs associated with having it actually administered in a clinical system. As I mentioned, the nurse, this is not kind of part of their routine, spectrum of knowledge and work for postpartum nurses. And so we've had to train them up, and even then, there are still some gaps in their knowledge that we continue to work with. And then management of the vaccine stock. So this is actually tied to this issue up here. Our current workflow is a little bit cumbersome. It involves printing the order, signing the order, faxing the order, and it's because it's a pilot program, it's being monitored by investigational drug services, even though it's not an investigational drug. On the facilitator side, so things that have really worked for us, we had some really great colleagues on the pharmacy side who have been our champions, who have really facilitated us even being able to get to this point and collect this pilot data. And we would still be in that year-plus process if it wasn't for them. On the inner setting, on the postpartum side, as I mentioned, nursing does have experience with other vaccines. And so that has been very beneficial in kind of getting them up to speed. We had a lot of hands-on intensive educational sessions. There are providers. We had also, I didn't share it here, but we had also done some more quantitative surveying using implementation tools, and our providers were ready to go. They were on board with this. And then we did a lot of using qualitative improvement tools, these rapid cycle, after every patient that was eligible, kind of assessing the situation, trying to work out the kinks. And that created for a very smooth implementation process from the perspective of nurses and clinicians, which they were very happy with. They said it's probably one of the smoothest rollouts of a new program they've seen. So I think that was really good. In summary, we need any novel strategy we can come up with to improve catch-up immunization. Implementation science is a methodology that could be used to better integrate those types of strategies into routine care and bring it out of the research world. And this particular program we found is feasible here, and if we continue to address the obstacles to its implementation, then it may facilitate broader adoption of this type of strategy, and it may be a highly effective and acceptable way to complete the vaccine series in this postpartum population. We have to continue doing our data collection on this side. We haven't done any qualitative work with the providers yet. Future studies are to just continue to fully integrate this program into routine care, both here, but I think there's opportunities to do this, at hospitals all over the, potentially making this type of program available to other inpatient populations, namely pediatrics, to really get to the targeted group of 11 to 12-year-olds. And then to utilize similar implementation strategies on the outpatient side. I want to thank both several mentors from the research aspect of this, but also several people from women's services and pharmacy that have really helped to get this pilot program off the ground. And this is the kind of patient handout that we've been using with fact sheets on the back. Thanks.